Live, Christian.
We're good.
Yep, we're good.
All right, cool. All right, good afternoon, everybody. Welcome to ULRI Research Institute's Chemical Insights and our webinar series. Today, we're going to learn a little bit more about the computational toxicology capabilities within the Institute in order to advance chemical safety. During this webinar, I'd like to remind you that please to submit your questions through the Q&A box rather than the chat feature. And we will do our best to answer those either in real time throughout the webinar or at the very end. So thank you again for joining. All right, so for those of you who may know or maybe don't know, I joined Chemical Insights this past year in June. And what we thought we would do in this webinar series, at least take the first two webinar series and try to introduce you to some of the new exciting research activities within Chemical Insights, as well as give you a little bit of background on the organization itself, right? Many of you may not be familiar with Chemical Insights and a lot of the research we do. So I think it's important to at least highlight what our capabilities are in this effort and what we may be doing going forward. Right, we've undergone a pretty hefty series of expansions since the beginning or the end of last year, and I think I want to cover these type of expansion and some of those capabilities that we brought into the Institute in the first two webinar series. So the first one is covering those computational toxicology capabilities, and then the second one will cover a number of the more existing capabilities that were in Chemical Insights, preceding my arrival in June, as well as some of the capabilities in exposure science and analytical chemistry that we added since the middle of the year. Right, and so that's going to be our first two webinar series, and then we're going to go over in the last two webinar series, touch on some of the research that is ongoing at the Institute in chemicals of emerging concern in individual health, and then chemical safety and community health. Right, so please pay attention to our newsletter or website, as well as social media for upcoming dates on those different webinars. All right. So a little background of Underwriters Laboratory. Some of you may be familiar with the little UL logo that's on your toaster somewhere, right? But really don't necessarily know or understand where we came from and how this all began, right? The UL legacy began when an enterprising young engineer named William Henry Merrill decided to take that step around the Chicago World's Fair. That was about the time when America was becoming electrified, and the insurers of the Chicago's World's Fair were concerned about underwriting the World's Fair because they were worried of the chance of a fire and it burning down. So what William Henry Merrill did was to develop the science and the tests that were needed to assure the safety of the electrical equipment around the World's Fair, and gave confidence to the insurance industry that yes, that the World Fair wouldn't burn down, and the overall the people that would be attending the World's Fair would be relatively safe. Right. The next year, Underwriters Electrical Bureau was founded with a small laboratory above a fire station. So to his credit, he decided to take that effort that he started in the Chicago's World Fair and turn it into a nonprofit research and testing organization in the early 1900s. And it was incorporated as a nonprofit after Merrill partnered with another engineer to expand into fire suppression and other safety-related areas. So UL has a really long legacy of safety-related both research and testing related initiatives. Next slide, Sarah. Since the early 1900s, the UL enterprise has evolved considerably, right? Through that time, we actually split up into three separate organizations, all of them rooted in safety science. In 2012, the organization was first broken up to spin out to the UL Solutions Enterprise. That's the primary for-profit entity that's involved in testing, inspection, and certification. So they do that in a for-profit way and for fees that are generated for testing different equipment or products for their safety based on certain standards that are set for that testing, right? Um, then in 2021, the organization was further split into two nonprofit organizations. One is UL Standards and Engagement. That's a 501c4 organization that really develops those standards to which UL Solutions tests to, as well as does stakeholder engagement, sort of promoting those standards much more broadly. And then we have UL Research Institutes. That's the 501c3. It's a full nonprofit public charity that's stayed since that kind of founding in late 1800s and early 1900s. And it really focuses now on research and education focused initiatives with a safety science focus, right? Uh, so that's kind of the background of both the UL enterprise as well as where Chemical Insights comes from. UL Research Institute is comprised of 6 different research institutes covering different aspects and facets of safety. What you see here is Chemical Insights Research Institute. We're located at 2 sites, and we'll talk about that in a minute, both in Marietta, Georgia, and in Research Triangle Park, North Carolina. We have a Digital Safety Research Institute that's virtual and distributed throughout the U.S. We have an Electrochemical Safety Research Institute focused primarily on battery safety and other electrochemical processes in Houston, Texas. We have a Fire Safety Research Institute in Columbia, Maryland, and an Institute for Research Experiences and Education, and that's also virtual. And lastly, we have a Materials Discovery Research Institute that's located in Skokie, Illinois. And all told, we have about 200 researchers that are all part of those 6 different research institutes. Next slide. So what is Chemical Insights and how do we fit into the larger chemical safety research activities within ULRI? Well, what we do is really provide unbiased research on chemicals and the risks to people's health, right? What we're driven to achieve is that we want to be the most innovative go-to research organization for trusted chemical toxicity and exposure information. And Katie and Steve are going to talk a little bit more about some aspects of how we do that research, right? And last is what makes us different, right? Why are we different from many of the other research institutes or academic institutions or even government research organizations that are out there? Well, we're a independent, nonprofit research organization, and talked about that legacy. And we build upon that over 130-year legacy that we've developed when Henry Merrill started the organization, and our goal and our mission to ensure public safety. So that's really what makes us different from a number of the other research institutes and organizations and academic institutions out there. Next slide. Well, right now we have, as I said, the Institute in 2 different locations. One is the original location that's located in Marietta, Georgia, and historically we focused predominantly on research that involves outdoor and indoor air pollution. We also have a research portfolio that you'll hear in the next set of webinar series about vaping, also about fires at the wildland urban interface, as well as specific fires around products such as the battery fire emissions, which is a public health concern. And then also different research initiatives associated with consumer product exposures. Right? Together, we developed a separate site at Research Triangle Park, RTP, North Carolina. We added capabilities in computational toxicology and exposure, high throughput in vitro testing, non-targeted analytical chemistry, as well as software development and data management tools. Right? And we believe that together, these 2 integrated type of research activities fill a pretty broad set of capabilities that allow us to address a very set of complex research questions within the chemical safety space. On the left-hand side, we're able to evaluate more integrated chemical exposures on products that consumers and the public may be exposed to or use on a daily basis, as well as environmental exposures in emergency and well-type fire scenarios. On the right-hand side, we're combining that with more rapid ways to evaluate the safety of these different chemicals as well as integrated exposures, as well as characterize what those exposures may be through those non-targeted analytical chemistry type of methods. And then lastly, we're developing capabilities that take all the data from those higher throughput as well as integrated testing type of methods, manage that data, and present that to our stakeholders through different means in a way that can inform decision making in multiple different levels, both at the consumer level as well as through companies and regulatory type of decisions. Right. Next slide. So, what I promised today is that you're going to hear more about the capabilities that we've been standing up in Research Triangle Park, North Carolina. You're going to hear first from Katie Paul Friedman from the new Center for Chemical Informatics and Screening. And then after her, Steve Edwards is going to come and tell you about some of the newer capabilities we have in the Center for Scientific Computing and Data Management. And with that, I'm going to turn it over to Katie, your first up, and why don't you take it away?
Thank you, Rusty. I really appreciate the introduction and the opportunity to speak with everyone today about our new center. Next slide, please. The primary problem facing the Center for Chemical Informatics and Screening is the too many chemicals, not enough information problem. Chemicals are necessary for our global economy, but their production and release clearly exceeds traditional safety assessment capacity. Estimates of the global chemical inventory from 2020 approached 350,000 chemicals, with 70,000 of those registered within the preceding 10 years. This really reflects the fact that global chemical production continues to grow, doubling from 2000 to 2020. Indeed, investigation of chemical pollution, dubbed as novel entities, suggests that chemical release may be approaching what's known as a planetary boundary or some sort of carrying capacity for sustainability on Earth. And yet, we know so little about these chemicals from a safety perspective. According to work led by Dr. Kristen Isaacs, who is also now with Chemical Insights, fewer than 15% of chemicals in US commerce have traditional data on use, emission, or exposure. And similarly, traditional hazard data are also lacking. For example, of the nearly 30,000 chemicals on the Non-Confidential Toxic Substances Control Act inventory, only about 10% have any traditional toxicity data, and less than 3% have a human health assessment. So we might step back and ask, why do so few chemicals have traditional assessments? Next slide, please. The answer is intuitive to all of you, I would imagine. The processes that we have developed and that we trust require too many resources and too much time. Typically, animal studies, including a range of exposure durations from acute to chronic, as well as specialized designs, are employed in a battery. And then the apical endpoints from these studies are measured, and the dose-response relationships for adverse effects representing toxicity are evaluated. Multiple studies in a battery are reviewed, and in some cases, systematic literature review and synthesis are also needed to develop these really thorough human health assessments. This process takes a variable amount of time, from a few years to, in some cases, in excess of a decade for a single chemical. In addition to the time, the cost per chemical is high, depending on the regulation, the production volume, and the studies that might be required. This can cost upwards of $1 to $5 million for a new higher tonnage chemical. Further, that process here has inherent uncertainty and limitations, which to date have required expert judgment to review. So clearly, this process is not going to be used to evaluate hundreds of thousands of chemicals. And you might ask, what happens if the last chemical we ever evaluated was actually the worst one? Next slide. So if we examine the human health toxicity testing paradigm in a modular way, we can view a new approach methods or NAM-based process as running in parallel to the traditional one, as in this figure from Brown et al. In 2024. The traditional approach, which is on the left, typically combines systemic in vivo toxicity tests along with those that are specific to a toxicity type, such as reproductive or developmental toxicity. This battery of tests is integrated and evaluated for nonspecific effects and specific effects. But many chemicals may only demonstrate nonspecific effects, such as decreased body weight or signs of systemic toxicity related to a target organ system or a maximum tolerated dose. And in some cases, the distinction between specific effects, such as effects on development, and nonspecific effects may be confounded or difficult to disentangle. However, a primary feature of this process, this human health assessment process, is to find a dose without appreciable risk to humans, a threshold dose, and then to further ensure that the assessment is protective for chemicals with any specific effect. And on the right, in a NAM-based toxicity paradigm, this is exactly the same, mimics the traditional pathway, where you'd have broad coverage models, i.e., a battery of in vitro models that cover sufficient biological breadth, paired with target-specific batteries. And together, these form an integrated dossier of NAMs for systemic toxicity and NAMs for other targeted toxicities that could be developed, leading to a protective bioactivity-based assessment, as well as targeted assessment of specific effects as needed. And we can note several points here listed to the right. A protective, nonspecific point of departure from animal studies or from NAMs are both consistent with public health protection goals, and setting limits for allowable exposure based on a bioactivity threshold is generally protective of human health. With additional testing, it might be possible to derive sufficient information on chemicals that have nonspecific effects to then predict some relevant mechanism affected by the chemical. But importantly, using NAMs, we can create NAM-based points of departure for hundreds or thousands of chemicals. This is the clear distinction, and this is public health protection at scale. Next slide. Our most important resource for creating those protective human health values for hundreds to thousands of chemicals is the team of people that we are building in the Center for Chemical Informatics and Screening. Our cheminformaticists, bioinformaticists, and high throughput screening scientists work hand in hand to create NAM products for chemical safety. And we continue to grow. We've just hired a broad profiling scientist, and we are actively hiring a chemical library manager and a computational toxicologist. Next slide, please. Our cheminformatics capabilities encompass chemical curation, defining chemicals of interest for study, developing and expanding databases for chemistry and properties to support improved downstream modeling, and the predictive modeling of chemical properties, including hazard properties. In terms of bioinformatics, we're working on bioactivity and traditional hazard data analysis, development of standardized computational workflows, and integration with exposure scientists and toxicokinetic modeling to understand dose and priority, and ultimately risk assessment. In our high throughput screening laboratory, we are establishing a number of capabilities, including in-house chemical library management and the use of specialized equipment to ensure high density screening, including both broad profiling and targeted screening. And then there's the integration. Can you step back a slide, please? Then there's also the integration of these capabilities working together, working with software and scientific computing scientists, as well as other toxicology, exposure, and analytical chemists, which will increase our cross-discipline impact. Next slide, please. And together, we intend to resolve the gap between chemicals with human exposure potential and the number of chemicals with hazard information. This work can be categorized into three primary areas of building. One, the curation of existing hazard information combined with systematic computational interpretation of these data, resulting in hundreds to thousands of toxicity values for different exposure routes. Two, development of standardized methods for in vitro data generation, both for systemic toxicity and targeted toxicities, resulting in empirical values to inform NAM-based assessments for thousands of chemicals, and with the power to really change the chemical assessment paradigm. And three, cheminformatic approaches to develop models for chemical hazard properties, thereby extending what we know about chemicals using in vivo models and in vitro models, and further resolving the chemicals for which data need to be generated because they fall outside of our structure-based inference space. Next slide, please. For rapid human health toxicity values, we have some choices for addressing the hazard gap. First, database calibrated toxicity values can be estimated from curated hazard data that already exist. A systematic approach to converting database information to toxicity values overcomes the need for expert knowledge and dramatically reduces the time needed to develop human health toxicity values. Second, in the middle, in vitro data can be used to develop new approach methods-based PODs, or POD-NAM, for data-poor chemicals. And given the many technical choices that can differ across early implementations in the field for POD-NAM, this requires some standardization to build trust that anyone can create or reproduce these empirical values. And finally, in silico inference using quantitative structure activity relationships. This can extend existing toxicity values, calibrated toxicity values, or in vitro-based toxicity values, and extends the data we have to the greatest number of chemicals possible. Next slide, please. For in vitro-based assessment, published frameworks in the literature do vary, as I mentioned, but they generally incorporate some key steps. First, collecting existing information, underscoring the necessity for developing, maintaining, and growing the best databases for toxicology possible. These existing information can include exposure scenarios and estimated total exposure, hazard information, if any exist, and any chemical grouping information on the basis of structure or properties. Second, many paradigms combine in silico and in vitro approaches to understand key properties or potentially off-ramps from performing an in vitro-based assessment, such as performing a read-across instead. And third, for data-poor chemicals within domain, the generation of the in vitro information itself of a sufficient biological breadth to estimate a POD-NAM. This can be pursued and potentially combined with select targeted NAMs. And then fourth, using these in vitro data along with toxicokinetic data and models to estimate the human equivalent dose corresponding to some bioactivity threshold. Next slide, please. Importantly, POD-NAM can be developed for hundreds to thousands of chemicals screened in high-density formats like 384-well plates using a battery of assays, potentially including assays for specific protein or pathway disruptions, high-throughput transcriptomics, and high-throughput cellular profiling assays that evaluate cellular morphological changes. The concentration-response relationships can be evaluated, and toxicokinetics can be applied to estimate that human equivalent administered dose corresponding to a threshold for biological disruption, regardless of a potential apical outcome. This process can be standardized to reduce resources and expert judgment, such that POD-NAM can be generated reproducibly at scale. Next slide, please. To bring this to reality, we are developing a physical laboratory to partner with our computational laboratory. The physical laboratory in Morrisville, North Carolina, will contain specialized equipment to enable this entire workflow from chemicals to handling to screening, all the way to integrated computational data analysis. And this lab will be mere feet away from our computational collaborators. Next slide, please. Our first aims are to create and implement standardized methods for common data requirements in human health assessment. When repeat dose toxicity or reproductive and developmental toxicity information are required, currently a commonly accepted alternative is read-across or grouping, typically on the basis of structure. We are working on additional in silico and in vitro NAM alternatives, which would also increase the number of chemicals with these types of information. We're developing database calibrated toxicity values and predictive models, as I mentioned on a previous slide. And we're developing standardized implementations for POD-NAM and NAM-based endocrine and developmental toxicity screening. Next slide, please. In conclusion, our computational toxicology capabilities are designed to produce human health information for thousands of chemicals on a rapid basis. At the foundation of everything we do is cheminformatics, and a defined chemical landscape to support all of our downstream activities for human health safety assessment, including data curation in collaboration with our sister center for scientific computing and data management, data generation in our high throughput screening laboratory, and standardized computational methods to interpret both the curated and the generated data. And ideally, these processes feed back to each other so that we are always focused on the chemicals of greatest interest for information gathering. These activities yield solutions for human health safety assessment, including some of the products that I've talked about in the preceding slides, like toxicity values, read across, predictive models, and NAM-based assessment. Our impact requires scientists across chemical insights to help with translation of our products and tools. And next, Dr. Stephen Edwards, the director of the Center for Scientific Computing and Data Management, is going to tell you more about our multifaceted computational toxicology work with an emphasis on how we create a platform that enables us to grow our data and tools, ensure public accessibility, and the opportunity to translate our work to the public.
Thank you, Katie. And I am looking forward to talking to you about the capabilities for our new Center for Scientific Computing and Data Management. Next slide. So I've broken the capabilities into two large groups, which are intentionally named very similarly. The computer science side is more your traditional software development, data engineering, and information technology, whereas the scientific computing is really understanding the data and trying to interpret the data and work with our subject matter experts for deeper understanding. And next slide. The advantage we have is that all of our folks are cross-trained in these different areas, but they come from different backgrounds. So we have a full range from people who started on the computer science side and all the way to people like me who started out in the laboratory doing experiments in the cold room. But the nice thing is we all bring our own unique experiences to the problem, but we all understand the common language. We also are a small team, but we are growing, and we would love to see some of you join our team in the coming weeks. Next slide. So our ultimate goal is to expand the impact of Chemical Insights data. And to discuss this, I need to rewind back to the turn of the century, where most scientific data were being released through scientific publications. And it looks like our animation okay. Looks like our animations didn't work, but that's okay. So if you look down in the bottom right, let's see if I can get a pointer. Okay, hopefully folks can see the pointer. So down in the bottom right, the scientific publications were typically used, and this. Oh, okay. Now I see a pointer. The scientific publications were primarily used, but it was a limited audience that could use those scientific publications, and the data were summary data that were coming out of those. As online data journals became the norm, the authors would publish their supplemental data along with those publications, and that improved things a bit. As the data sets got larger and larger, we would provide downloadable files either through public repositories or as the organization themselves. But those data sets required a the user to develop processing pipelines that could process those data. In parallel with that, along the top were websites that could allow users to interact with the data without having to develop these processing pipelines. And while those websites initially were focused on the scientist, in theory, if those websites are developed appropriately, this could make data accessible to everyone. Of course, we're not limited to data generators building websites. We can have third parties that develop websites based on the data coming out of those different places. Early on, again, that would require processing of the data by those tool developers. But as data become available through application programming interfaces or APIs, we can actually facilitate the development both of third-party tools based on data, as well as additional research that builds on those data without requiring the researchers to go all the way back to the raw data files. As far as the audience, okay. Yep. As far as the audience, we expect that the websites would gather the most people. So that's the little blue buttons up in the top right with the loud sound. The application programming interfaces kind of represent a step down where people can access the pre-processed or the post-processed data more readily. So it's a smaller group, but larger than the group down in the bottom left, which would be starting from the raw data and building their own processing pipelines. But all three of those mechanisms of data dispersal are important, and so we want to support all of those at Chemical Insights. And I do want to point out back down in the bottom right that even though the traditional publication method is still a limited impact from the number of users, it provides the scientific credibility for everything else on the slide. And so it really is the cornerstone on which everything else is built. And now we can move to the next slide. But disseminating the data is only half the story. For the data to have an impact, we also have to ensure that the data is of the highest quality and also that the data is usable. For the usability aspect, we're going to be using the FAIR principles that were released in 2016. So they're 10 years old now. And that stands for findable, accessible, interoperable, and reusable. So we will be using those guiding principles to make sure our data are usable, and we will be employing other techniques to ensure the highest data quality. We have to do this through three different means or through three different data types. So there's the data that you heard about from Katie that we will be generating, and you will hear about on the next webinar, that we will be generating within the Chemical Insights Institute. There's also structured data from other sources that we would be pulling in and integrating with our Chemical Insights data to make a larger corpus. And where data gaps exist, we will be going out and seeking data. You heard from Katie about the database calibrated toxicity values. This requires us to extract and standardize those data from other sources. Next slide, please. So our internal data is the easiest to work with. We can, because we have ultimate control over the entire process. So we can collaborate with our lab colleagues to make sure that the quality assurance procedures are well documented, and we can work with them to build quality control tools that help capture errors when they happen and before we release those data to the public. Most of the time, the appropriate metadata are collected. They just don't follow the data as it progresses through the different stages. And so that's the main thing we'll be working with, and we'll be leveraging LIMS systems and other means to make sure that the metadata that our laboratory folks are collecting along with the data follow that data all the way out to the ultimate users, so they have that available when either processing the data through their own processing pipelines or just doing a data analysis so they understand the underlying assumptions. We'll be using standardized languages both to help with our own internal integration efforts, but also to ensure interoperability with other groups. And there's several other points towards the bottom that that also help the fairness of our data. Next slide. So as I mentioned on the previous slide, the chemical insights data is the easiest to work with because we have complete control. Public data repositories are really helpful because they typically have done a lot of the standardization and integration of the data themselves. And so we're simply trying to coordinate our data with their data and integrate from there. However, most of those public data repositories are secondary sources, and so they lack a lot of times the full data provenance. And so we will then have to go and find the primary sources and include that documentation if needed. The data from the public documents, on the other hand, typically are the primary data source, so we can gather a lot of those data directly, but they're not typically designed for this type of data integration dissemination. And so we end up having to do a lot of work to harmonize those data and integrate them with our other data sources. Next slide. And so making those data available are a value in and of themselves, but certainly we want to push further. And so the metadata standardization that I've discussed on the previous slides can also allow us to do work in data harmonization, as well as quality tagging and weighting of data based on the quality metrics. And that allows us to provide ever larger data sources of quality data, garbage in, garbage out, that can be used in building predictive models. And those predictive models then can be used in developing lots of decision support tools. And so you can see how at each step we're magnifying the impact, because one data set now can facilitate multiple predictive models, and each model can then be integrated in multiple decision support tools. Next slide. So now if we loop back to our ultimate goal, we want to make sure that we're providing data in a form that all users, whether it's a casual web user or a person who wants to build a new processing pipeline and explore science that maybe we haven't even thought about before. We want to make sure that all of those users have the data in a format that they can use. We want to make sure that that data is of the highest quality possible. And we want to make sure that we've implemented our FAIR principles such that the data are usable and interoperable with other groups. And in doing so, we anticipate that we will be able to develop many web tools to help the public, as well as other groups that rely on our data. And so eventually, we would like to not only facilitate the decision makers, within government entities or industry, but also the individual person in the public who wants to make their own personal decisions about the safety of the products that they use every day. And so with that, I'll hand back to Rusty and we can take questions.
Got unmuted. So thank you, Steve and Katie, for those excellent presentations. We have a number of questions in the chat, so let's start at the top. Katie, I think the first one's for you. Says, could you please expand on your approach on using POD NAM values for risk assessment? Can you provide some examples?
Yeah, that's a great question. So in terms of using a POD NAM value in risk assessment, I'd say this is a piece of the puzzle. When you have data poor chemicals, depending on the regulatory environment that they're in, you need a place to start in terms of a conservative dose estimate that would be without appreciable risk to the human population. And so that's really the piece of the puzzle that POD NAM could resolve for a lot more chemicals at scale. But it's not the only piece of a human health assessment. And so, depending on the regulatory framework, the tonnage of production, what geography you're in, there may be other data requirements that would be needed. And so, POD NAM is that first piece of a quantitative risk assessment that then could be added to depending on the specific requirements for that chemical and the space it's in.
Awesome. Thanks, Katie. Next question is, I'm mostly worried about the conservatism in broad terms built into bioactivity-based compared to AOP-based threshold values. How do we envision to refine general bioactivity approach to give more realistic but yet still protective values?
That's another great question, and Rusty, I'm sure you have thoughts on this as well. But in terms of developing a POD NAM, you have your in vitro assay technologies, which we know can produce a protective point of departure. I've published on this previously with various groups, but other groups have published on it as well. Um, but then, you also have an element of how do you incorporate uncertainty into the derivation of a toxicity value. So in a traditional process, you would have a point of departure, and then you would develop a reference dose or toxicity value based on that original point of departure. So the ways in which we incorporate uncertainty into developing a toxicity value based on a POD NAM could actually take into account maybe some of the conservatism or different ways that in vitro NAMs handle uncertainty. And sometimes that uncertainty is a bit more empirical, and we could incorporate it to get a different kind of reference value. Another way to answer that question might be in terms of, you know, AOP based points of departure, because we don't necessarily have AOPs that address uh, systemic toxicity or actions of chemicals that are not fully characterized. I'm not sure an AOP based approach would be broad enough to get you the original point of departure, but the AOP approach could help you refine what additional data you might need after you interpret the data that are generated for a POD NAM.
The thing that you may want to add, Katie, or elaborate on is some of the study that we were co-authors on comparing more of the bioactivity-based points of departure with the human clinical studies, in addition to, and also comparing them with more traditional animal studies. So do you want to elaborate on that a little bit?
Sure. Yeah, we can go in that direction as well. So, Dr. Chelsea Weidekamp, who is one of the scientists in our center, she's the lead author on a paper that Rusty and I and others co-authored, looking at the sort of quantitative concordance between in vitro, POD NAM, and in vivo clinical data for drugs, where you actually have this information. For environmental chemicals, you typically don't have the human data to compare to at scale. And so when you look at comparing in vitro POD to human clinical points of departure, you actually have less of an uncertainty or adjustment factor needed to estimate the human point of departure from the in vitro POD NAM than you would using the rodent. To be conservative enough using the rodent, you actually have to divide by a larger composite uncertainty factor. So there are some ways of looking at this question of conservatism and whether or not animals are conservative enough for, the human response. And we inherently know that as toxicologists, right? Because we develop these composite uncertainty factors that you then divide the rodent data by. So that composite uncertainty factor might be different for a POD NAM.
They both are about the same, have the same concordance with human studies as well. So that's important to keep in mind. Another question is, are you hoping to ID these data poor chemicals through non-targeted methods as well as through self-reporting? That may be a question for next seminar, where I think Mark Wilson is going to talk about some of the non-targeted analysis capabilities in his center and how those are going to be used and fed into the larger research efforts that are going on in the Institute. So, but the bottom line is, yes, we are developing non-target analysis capabilities, and they will be used in terms of identifying some of the data poor chemicals that Chemical Insights may focus their research efforts on. So, hold on to your hats, and I encourage you to attend our next webinar on that topic. Another question is, are there cost savings using new approach methods over traditional animal-based methods? Katie?
Yeah, so if you use a traditional animal-based approach, as I mentioned in the slides, depending on how much data you might be required to generate, depending on how that chemical is used and what regulatory framework it falls under, you could spend $1 to $5 million to really generate the animal data necessary for a human health assessment versus a NAM-based approach. You're in the low thousands, depending on the battery that you build. You're, maybe 7 to 15,000 a chemical. So it's vastly different. There's a huge amount of savings. And the issue is, how to generate enough data across the chemical universe so that you would be able to extend inferences beyond even the data that we can generate. So I think the large cost of the in vitro data generation, we're often looking at it at scale. How do I generate these data for 100 chemicals or 1,000 chemicals? But if you look at it on a per chemical basis, the difference is vast.
Take the next one. So, is there any project to incorporate ecological toxicity values? And Maria, the answer to that question is not this time. The Chemical Insights is currently focused on more human health and not ecological based toxicity and values to inform ecotoxicity type of assessments. So, um, another question is, are you working in the domain of automating AI-driven toxicological risk assessment for the chemicals identified as extractable and leachable? Or do you want to elaborate on that one? Steve, I think AI is some of the effort going on in your group. You want to elaborate on both of you on some of those AI-driven risk assessment approaches. And you
Yeah,
can
I
talk
can
about
do that.
AI
So we're
being more of the broader, both machine learning as well as
Yep.
other large language model type of AI initiatives.
Yeah, that's exactly what I was gonna say is we're definitely looking to leverage the new generation generative AI approaches and integrate those in our tools, but we're not limited to that. So we're certainly, wanting to include everything from, structure-based predictive models to machine learning that incorporates more of the NAM information to, all the way along the continuum, including AOP based models where we have both the AOP and the data. So we're definitely looking to include AI where we can. Early on, I think the use of AI is going to be more upstream. So, facilitating the human element rather than having, AI that's directly making risk assessments. But it's certainly going to be a critical component moving forward, and I think, an ever increasing component as we move forward with the different automations.
Anything to add, Katie, or does that cover it?
Yeah, I mean, I think Steve covered it really well. Depending on how you define AI, some people do lump in machine learning, and obviously that is sort of bread and butter for some of our cheminformatic predictive modeling, to use machine learning in deriving those. But I think then integrating them into a platform, there's a lot, as Steve mentioned, that could be done to make it easier for users to interact with all of the different models. And there's some interesting examples now out in the field of people starting to do that and set up platforms for interacting with model outputs that have been generated.
Question is, do you adjust for metabolism when deriving a POD NAM? And how would you do that, Katie?
Yeah, thanks for the question. So I think the short answer is yes. One of the ways that you can do that is by incorporating metabolically active models into the battery of assays for POD NAM. And So that's been done to differing extents across the literature. Internally, we have Dr. Chad Dyson-Roth. So he has authored a number of studies on retrofitting assays with S9 for metabolism. That's one way of doing this at scale, but also incorporating models like various cell lines like HEPA-RG that might metabolize the parent chemical so that you can be sure to sort of capture that. The other end of metabolism, of course, is the toxicokinetics and how that chemical might distribute through the body and be cleared. And so that's a big part of translating POD NAM to a useful dose is taking into account that toxicokinetic modeling. And Dr. John Wamba is another scientist in our center who's been a leader for developing high throughput toxicokinetic models to do just that.
Katie Smith asks, great presentation so far. Will the data sources, computational methods, accuracy, and validation methods be transparent to the public? I will say first, it is a philosophy and commitment from on behalf of UL Research Institutes to have open science and transparency. So all of the work going on in all the institutes are open and transparent to the public. But you want to elaborate a little bit on that, Steve, in terms of what you talked about in terms of these the data being available through all the different modes that you identified in your presentation.
Yeah, absolutely. So definitely everything we generate internal to Chemical Insights, as I said, the intent there is to capture all of the information and have that flow out with the data so that you have the full provenance of that data. The data that we're pulling from outside sources, we're going to do that same thing to the greatest extent possible. So, the one exception to that can be with some of those secondary data sources if it's not possible through that to actually trace back to the primary data sources, then sometimes we have gaps in that information. But certainly to the extent that we can gather that information, which we will 100% do on Chemical Insights data, all of that will be made public through all of the different routes. So there's going to be complete transparency to the extent that we can provide transparency.
Thank you. Christy Sullivan asks, can you say more about the Institute's envision stakeholders? Perhaps provide some examples of how you envision the data you are generating being used. And Christy, I think we our different stakeholders, at least where Chemical Insights may vary depending on the project, right? We envision multiple different stakeholders. We have, as you'll hear in the next webinar series, we have vaping research where the research is really directed at the general public, right, to inform what are the health risks associated with vaping behaviors. We also have research going on where we're intending to, as Katie talked about, utilize the database calibrated toxicity values and use those to inform potential decisions either at the in different regulatory jurisdictions. It could be at the state level or the federal level or international level. We have other efforts going on to potentially transfer or develop these methods, and hopefully different industry will use those and adopt those methods to do product testing. So I think we have a broad range of stakeholders. But the bottom line is that we're doing this research in a very transparent, open way. And in a very unbiased way that can inform decisions at multiple different stakeholders and levels. So the next question is from Thomas Lane. How do you define chemical applicability domain, and how does this influence model prediction and confidence in your current pipeline?
Yeah, thanks for the question, Thomas. So there's a couple of different places, where we could talk about applicability domain. One is in that POD NAM space where a lot of the questions have focused and, what chemicals are actually logical to screen in different in vitro technologies. So, if you want to screen a semi-volatile or volatile chemical, you really need to capture an air liquid interface model versus chemicals that are of a sufficient molecular weight and certain properties so that they would be amenable to an aqueous-based assay battery, right? So So part of applicability domain for sort of next generation risk assessment is figuring out the battery and the pathway for your specific chemical. But if you mean more in terms of the cheminformatic integration and the model development, that's where that question gets very thorny and you get different responses across different cheminformaticists. I think it's still an area of active research, but of course, you can take a look at the development of QMRFs and the proliferation of those around modeling, and how do you describe essentially the set of nearest neighbors, and are you a chemical that's in domain because you are sufficiently close based on some arbitrary threshold for neighbors and things like that. And some of that might be model specific. So we could get into the weeds there, but it's a great question, and it's one that Nate Charay and Gabriel Sinclair are certainly encountering in our cheminformatics of how do we convey within these models whether or not a chemical is in or out of domain.
It looks like since we have about 4 minutes left, we won't get to all the questions, but we'll do our best, everybody, to answer them potentially later, typing them into the box. The next one's come from Shubham Nate. We are looking forward to working more in depth on chemical mixtures, mixture assessment factor regulations. Will this computational toxicology add more value by understanding the data for risk assessment on human health? This is your favorite
Um,
question, Katie, on mixtures.
Yeah, I told you mixtures were important. So, um, actually, this is an area that a number of us in Chemical Insights are pretty interested in from different perspectives. So I'll give you my perspective, which is, if we generate a lot of single chemical data and make that public, then it's possible to model those mixtures of those chemicals, to maybe prioritize chemical mixtures for further screening because there's n factorial mixtures you could possibly screen. That is still a problem, whether you're in traditional methods or new approach methods. The n factorial number of mixtures is daunting to address empirically. So, making single chemical data available for many chemicals allows you to make some simple assumptions, maybe concentration addition or independent action, and then simulate a number of mixtures that might be important based on human exposure. And that's certainly something that we are interested in.
Next question is from Sam Yoki. He's currently in his finals for a master's in environmental toxicology. He said, what's the biggest challenge in integrating in vitro data with in silico prediction? And are there any emerging trends in computationally you would like to highlight?
Oh, that's an interesting question. The biggest challenge in integrating in vitro data and in silico prediction, I might argue that what most data scientists, whether we're computational toxicologists or some other flavor, will tell you is that wrangling the data takes the largest amount of time. So the data curation and then wrangling it and understanding the metadata around it so you can integrate, that is always where we spend the highest amount of our time. So that continues to be a challenge. And I think hopefully 20 years from now, we will have some better standards for metadata and how data can interact more easily. And then the second part, emerging trends in computational toxicology. I guess I'll highlight the power of public data. I think at the beginning of my career in particular, a lot of people didn't want to share their data, sort of keep it behind a curtain. And working in this space has taught me that actually democratizing that data and allowing many people to use it in different ways is what has advanced us in computational toxicology. So that sharing and publication of data is just critically important.
I think that we have only a few seconds left in our time tonight, so I apologize for those that didn't get their questions answered. So, but I would encourage you to tune in in the next webinar series in Q2, a date to be announced to learn about the other half of Chemical Insights, which is really the Center for Toxicology and Human Health and the Center for Analytical and Exposure Sciences. Great two talks by Mark Wilson and our leaders for both of those centers. I'm going to really get going into depth about each of their research activities and capabilities as well. And Crystal Wright and Mark Wilson are going to be giving those two talks. So please tune in, and hopefully we'll see you next time. All right. Thank you all.